Hello and welcome. Today we're going to be having a discussion about ADCs. I'm going to be discussing considerations around stability attributes when it comes to developing these groundbreaking therapeutics. I will occasionally show slides in the media player to illustrate certain points, but otherwise you'll see a blank blue screen. So today I'm going to talk about ADC characterization. Antibody-drug conjugates, or ADCs, represent a growing share of the overall biologics market. And as they continue to be of interest to biologics researchers, I'd like to take today to discuss the considerations required to address their developability characteristics. We're going to start by talking a little about what exactly ADCs are for those who haven't worked extensively with them before. Then we'll dive deeper into the engineering and structural considerations of ADCs. And finally, we'll look at real data examples from the Prometheus Panta and discuss what parameters and attributes to examine when you're formulating your own ADCs. ADCs are exactly what they sound like, a monoclonal antibody that has been covalently linked to a small molecule drug. For the last decade, they have represented a growing proportion of the biologics market. And what's so interesting about ADCs is that they combine the targeting power of a monoclonal antibody, which is highly specific to a given antigen, with the druggability of a small molecule. This has the biggest implication on cancer treatments. Many of the small molecule drugs used to treat cancer operate by shutting down some aspect of cellular transcription or metabolism, thereby killing the cancer cells. However, they operate on a common principle. They must first kill the cancer cells before they kill healthy cells. The unfortunate side effects of chemotherapy are commonly a result of the chemotherapeutic small molecules also having a negative impact on healthy cells. However, ADCs offer an alternative to these highly toxic chemotherapies. Since monoclonal antibodies offer high specificity and they target only their intended antigen, we can target these to many cancer types. Since many cancer types express receptors that are unique to cancer cells, it is possible to tether the chemotherapeutic molecule to a cancer-targeting antibody, which essentially serves the cancer-destroying drug directly and only to the cancer cells. And this offers truly amazing potential as the next generation of cancer therapeutics. As with any biologic, there are many considerations around the stability, efficacy, and toxicity of ADCs. I'd like to focus today on their developability characteristics, most notably conformational and colloidal stability. And so to understand their stability, we first need to talk about the components of an ADC. So first, we have the monoclonal antibody. And this may be an already existing antibody with a known, well-characterized antigen interaction, or you may be working to develop a new monoclonal antibody for a new target or with different properties than originator. We'll talk a little more about why you may want to do that later on. Next, we have the small molecule drug. As with the monoclonal antibody, this small molecule may already be in use as a standalone treatment, or it may have been pulled from a synthetic library. It may also be built from a fragment library. And you may need to modify this additionally, essentially to prevent interference with the antibody or to provide space for the linker. And that linker is the last part of the ADC. It must attach the small molecule to the antibody in a way that the active part of the small molecule can access the target protein. Length of the linker and method of attachment to the antibody are critical considerations when building the antibody. So now that we've discussed the basics of ADCs, let's get into the practical considerations around building these complex therapeutics. While there are many different characteristics of ADCs to consider, for the sake of time, today we'll be focusing just on the developability characteristics of ADCs, mainly the conformational and colloidal stability. So there are many approaches to take for conjugation depending on where the linker is attached. I don't want to get too deep into all the chemistries and reactions available and how they work, but suffice to say each method has its own pros and cons, and the method you choose will likely be dependent on the particular small molecule you're adding and its own chemistry. But I'd like to use one example to illustrate this, as it will reflect the real-life data we'll view later on. And this is conjugation via amines. So amine conjugation must occur at a slightly basic pH in order to ensure the reduction of the lysine side chain. It's also important not to have any buffers with free amine groups, and this would include tris or glycine buffers. Reactions often have to incubate for anywhere between 30 minutes to several hours, and this means prolonged time in this environment. The conjugation process itself can be destabilizing to the monoclonal antibody, but longer incubations will increase the attachment of the small molecule. So already you can start to see that there's a balancing act between getting good reactivity and not destabilizing the monoclonal antibody to the point of it being denatured and losing its targeting ability. Additionally, it's worth considering the degree of conjugation when doing amine-based conjugation. The reaction will occur between any solution-exposed lysine and the reactive end of the linker. So we know monoclonal antibodies can contain up to 80 lysines per molecule, many of which are solution exposed. So this means that every individual molecule likely has multiple drugs conjugated to a single monoclonal antibody. But it also means that there's a risk of a drug binding a lysine in the antigen binding region, which can then reduce or even entirely eliminate the targeting power of your monoclonal antibody. We also know that bringing a small molecule close to the antibody will change its chemical environment, which can then have an impact on its stability. It's kind of like adding a buffer additive. It will have some impact on the antibody's overall stability. So even if the conjugation process itself is leaving the monoclonal antibody intact, there will be some impact to the quality attributes of this molecule. Of course, one way to reduce some of the impact of the drug on the antibody structure is by altering the linker length or choosing a linker with a different reactive site so you can use a different conjugation approach. Either way, once you've locked in the conjugation approach and the exact linker and drug to conjugate to your antibody, you must still consider optimizing its buffer formulation. And we know that a buffer that is stabilizing to a monoclonal antibody alone may not confer the same stability advantages to that monoclonal antibody once it's part of an ADC. And if you'd like to learn a little bit more about stability profiling for formulation, you can watch a short video entitled "How to Bind Identifies the Best Buffer for a Vaccine Antigen Target." And so with all of these considerations for just a single conjugation type, it should start to become obvious that the degree of complexity increases dramatically for an ADC versus a monoclonal antibody alone. So regardless of your method of conjugation, there are some variables you can tweak to reduce your risk of destabilizing the antibody. So first, you can determine if you have flexibility in the conjugation approach you'll use and experiment with alternatives. Second, you can formulate the naked monoclonal antibody in a buffer that is compatible with the desired conjugation reaction. And if that's not possible and you know the buffer will be destabilizing, you can try to reduce the exposure of the monoclonal antibody to the conjugation conditions. And finally, you can experiment with linker lengths, keeping in mind that the drug will act as a change to the chemical environment of the monoclonal antibody. So now that we've talked about experimental considerations, I'd like to talk specifically about how conformational and colloidal stability data, and here obtained from our Prometheus Panta instrument, can give you insight into how conjugation impacts your ADC and helps you optimize your therapeutic. So the ADC we'll be examining here is called trastuzumab emtansine, which is patented as Kadcyla. You may be familiar with Herceptin, which is a monoclonal antibody treatment for HER2-positive breast cancer. So this ADC is the Herceptin monoclonal antibody conjugated to a small molecule drug called DM1 that promotes cytotoxicity. And just for a little bit of housekeeping, since the drug names themselves are a little bit of a mouthful, I'll be referring to them by their brand names for the rest of the webinar. So this conjugation is a non-reducible thioether, and there are an average of 3.5 DM1 molecules per antibody. So the Herceptin-based antibody targets only the HER2-positive cells in a tumor, and then active DM1 release occurs only as a result of proteolytic degradation of the antibody. The DM1 triggers mitotic arrest and then eventually causes apoptosis of the tumor cells. So I'd like to emphasize that both Herceptin and the ADC Kadcyla are already available on the market. So we chose to investigate their critical quality attributes related to stability in order to show why it's so critical to have high-resolution multi-parameter data about their stability characteristics. However, if you're in early development, it's important to remember that these are antibody-based therapies that have already made it to market. They have ideal characteristics for stability and efficacy. So when you're working to develop these treatments early in the pipeline, you're going to see lots of bad candidates that need to be either eliminated or optimized. So first, we're going to look at the thermal unfolding profiles of both Herceptin and its ADC. And if you'd like to learn more about thermal unfolding, please check out the video called Thermal Stability Measurements for Biologics Developability Profiling. And very simply, we use the intrinsic fluorescence of a protein to follow its unfolding in response to a thermal denaturation gradient. And the Panta allows for very precisely controlled heat transfer to the sample, providing the highest resolution data. And so that's what this looks like. In navy, you can see the naked monoclonal antibody, while in blue, we see the ADC. The dashed lines here represent the inflection points of unfolding of different domains. Typically, you'll see the FC1 unfolding first, followed by the Fab region, and then the FC2 region. And often the FC2 and Fab regions have similar melting temperatures that cannot be resolved. So the second TM represents both of these domains unfolding. And then shown below is the first derivative view, which makes it a little bit easier to identify those inflection points. And just as a reminder, a decrease in melting temperature is indicative of decreased stability. So the data resolution here is very helpful, as it shows the degree of impact that the conjugation has on both TMs. But you can see that TM1 is decreased by 2.5 degrees Celsius, while TM2 sees a more modest decrease of 1.4 degrees Celsius. Now, these experiments were run in triplicate, and it was determined that both temperature changes were statistically significant. So the more dramatic decrease in melting temperature for TM1 suggests that the majority of impact from the conjugation was had on the FC1 domain. And this is preferred as the Fab domain contains the variable region necessary for antigen binding. Now, I'd also like to point out the peak broadening that we see only for TM1. The first derivative peak is wider compared to the naked antibody, and in the ratio view, the T onset decreases to a greater degree than the TM. This is also a sign of destabilization of the ADC, and likely due to the presence of the DM1 molecules linked to the antibody. They interrupt the chemical environment of the antibody and cause it to be less thermostable. We know that the degree of peak broadening may also be impacted by the number of molecules attached to the antibody. Anywhere from 0 to 8 are attached, although we know that 3.5 is the average for this ADC. So the conformational destabilization from the drug conjugation is significant. However, the decrease in TMs and even T onset is modest. Obviously, it's best to avoid destabilizing your antibody, but this is a good example of minimal changes to the thermal stability profiles. It's basically going to be impossible to have no stability impact from drug conjugation, so this is definitely an ideal scenario with only a few degrees drop in stability. But we know that thermal stability is not the whole picture. So here, we're looking at the colloidal stability information of the same samples. These were derived from back reflection turbidity measurements as well as light scattering optics. The Panta is the only instrumentation that allows you to collect all the stability data in parallel along a thermal denaturation gradient. And if you'd like more information about how these measurements work, you can check out the Explore Prometheus Panta video. So turbidity measurements reveal large amorphous aggregates, while light scattering measurements are more sensitive to small changes in the size of your molecules. And together, these unveil the complex unfolding and aggregation behaviors of your samples. So first, you can see that the large amorphous aggregates from turbidity do not appear until the unfolding of TM2. But now I'd like to focus on just the scattering and turbidity profiles so you can see the changes more closely. So scattering shows size changes associated with the unfolding of the FC1 domain, that is TM1. However, there's not a huge jump in scattering, which implies that the domain unfolds but does not aggregate, as you can see here indicated by the arrow. You can note that the turbidity and scattering profiles of the ADC are fairly similar to the naked monoclonal antibody, but they also exhibit earlier onsets of turbidity and scattering compared to Herceptin alone. And again, I'd like to emphasize that these are both biologics that have reached the market and been optimized for the clinic. So the ADC has minimal decreases in stability compared to the Herceptin, which is what anyone working to develop an ADC should strive for. So to summarize, the parameters to optimize when looking at a thermal denaturation ramp are a minimal decrease in your melting temperature for all domains, no peak broadening in your melting temperature, minimal decrease in other parameters, including T onset, T turbidity, and then T scattering or T aggregation, an overall similar profile shape, and no increase in scattering, as this would imply contaminants, unconjugated drug molecules, or unfolded sample at the start of the experiment. So next, I'd like to discuss more about colloidal stability. And here we're going to look at an isothermal size distribution experiment to show how conjugation impacts the size and dispersity of your therapeutics. And so this is data derived from DLS, which informs on the size of the molecules you've got in solution, as well as the distribution of particle sizes. Isothermal DLS measurements are supercritical for determining whether there are contaminants, unconjugated drug, or aggregates in your sample. This measurement was performed at 25 degrees C before the thermal denaturation gradient we saw earlier, and it's a reflection of how your biologics are behaving on the bench. And for a deeper dive into how the DLS data are generated, you can watch the video titled DLS Easily Explained. So as you can see, the overlay and distribution between the naked monoclonal antibody and the ADC are very similar. There's a very slight change in the hydrodynamic radius associated with the conjugated antibody of 0.1 nanometers. And this is not statistically significant. because DLS cannot always resolve the sub-nanometer differences associated with the hydrodynamic radius change from drug conjugation, but it does show that there was some overall change to the structure of the molecule. And what's also important to note is that the peak does not broaden. Peak broadening for intensity distribution profiles implies destabilization, as there are multiple populations close to, but not identical in size and present. That is, if your peaks are broadening, there are slightly unfolded, but not yet aggregated or fully unfolded particles in your mixture. Further, there is a very small peak at a high hydrodynamic radius, as indicated by the arrow. Larger sized particles are overrepresented in intensity distribution plots. However, it's noteworthy because there's no corresponding peak for the naked monoclonal antibody. Last, I'd like to remind you that if your conjugation process does not completely use all the small molecule, or if you don't have sufficient cleanup process after the conjugation, DLS can resolve particles as small as 0.5 nanometers in size, which means they will likely pop up as an additional peak on the graph, and that would be here. And so we don't see that here, but it is worth considering during your conjugation. So overall, there's not a huge difference in the DLS profiles of the ADC versus the Herceptin, and this is a good thing. As with the thermal stability profiling, it's expected. Both of these biologics have been optimized for clinical use. However, these do give you an indication of what to look for during the discovery and development phase. And these parameters include no significant change in hydrodynamic radius, no peak broadening, which would indicate non-uniform particle folding, no significant size population at low hydrodynamic radius, and it would be indicative of free unconjugated small molecules, and no large aggregate populations. Another stability characteristic you can investigate with the Prometheus Panta is chemical denaturation. This is a great conformational stability indicator that tells you about the free energy of unfolding in isothermal conditions. So to set up a chemical denaturation, a gradient of chaotrope such as urea or guanidine hydrochloride is added to a constant target concentration of the protein of interest. Now, once the unfolding equilibrium is achieved, the Panta uses the same intrinsic fluorescence from nano-DSF to look at how the protein unfolds in response to increasing concentration of chaotrope. And with that information, we can calculate the delta G, or the free energy of unfolding. This parameter is also helpful for ranking chemically stable candidates, as a higher delta G requires more input of energy to unfold and is therefore more stable. So both the antibody and ADC denaturation are fit with a three-state model, as, similar to thermal denaturation, the different domains are impacted separately by chemical denaturation. We can see the first delta G for the ADC is decreased compared to Herceptin alone. This goes from about 26 to 21 kilojoules per mole. For delta G2, there's a more dramatic decrease from about 81 to 71 kilojoules per mole. So if you choose to investigate the chemical stability of your ADCs during development, you want to look for minimal impact to the delta G. So ADC developability assessment requires collecting information about many of the same critical quality attributes for monoclonal antibody development, but both the conjugation process and the drug itself will add complexity to their analysis. So for ADC development, it's important to fully characterize both the individual components, the drug and the antibody, as well as the final conjugated product. And of course, there are many more attributes to assess outside of what we talked about here today. But the really great thing about what I've shown today is that it was all done on the same instrument, and much of it was done in parallel on the same sample. So thermal stability measurements tell you about conformational stability and enable you to compare how the conjugation process impacted different domains of your ADCs. Colloidal stability measurements will tell you about aggregation and whether your cleanup process was sufficient. So with that, I'd like to thank everyone for joining us today to discuss some of the many critical stability attributes to investigate when developing ADCs. And I'd like to give a particular shout out to my colleague, Dr. Andreas Langer, who worked behind the scenes to collect all this data and so much more that we unfortunately didn't have time to show so that we could share it with you all today. So thank you to Andreas.